Okay, welcome everybody to the webinar Organ on a Chip Meetup, a preparation for Organ on a Chip Masterclass. We are super excited to have this webinar because around 2 weeks we are gonna have the masterclass, a masterclass that's organized by ASA Innovations and ICAT, and you see also the participating companies, which we are gonna hear from them today as well. It's a full 3-day program. Really, I find it really cool. I'm one of the trainers together with Emre and together with the amazing people at ICAT. We are organizing this. So maybe we can go to the next slide to kind of show the agenda of the webinar. We are going to show you how the masterclass looks like, who we are, who ICAT is, and how the masterclass looks like. And then we are going to focus on the technologies, amazing technologies by the companies. We are going to hear from Proefdiervrij and their activities. And then we are going to hear from STEMCELL technologies, because part of the training is an online training by STEMCELL technologies on culturing organoids. And at the end, we are going to have an FAQ, most common questions, but also if there are more questions of anybody here, we can answer at the end. If you have questions, again, put it in the Q&A or the chats. And then if you need to ask the questions yourself, raise your hand, and then I'm gonna enable your microphone or camera so you can ask the questions from the presenters directly. Okay, I think I've talked enough. I want to give the floor to my colleague Emre to talk a bit more about other innovations and the masterclass. Emre, floor is yours.
Yeah, thank you very much, Hossein. So my name is Emre. I'm the colleague of Hossein at ASA Innovations. And at ASA Innovations, we're passionate about helping scientists to adopt this wonderful technology of organ on a chip. But we also know that it's sometimes complicated to do so because it can be quite a hassle to implement the organ on a chip system. So that's why we like to help you. And we do that through research and development projects, through consulting, but mostly via education. And that is also what the masterclass is about. So I will give now a brief overview of what to expect from the masterclass, because some of you might have already signed up, or some of you might be interested in signing up. So hopefully this will give a nice overview. And if you're interested in signing up, I would like to bring to your attention that the deadline is 24th of September. So hurry up, because there's also just a few spots left. And hopefully see you on the 1st, 2nd, and 3rd of October in Utrecht. So this is a picture from last year. As you can see, lots of happy faces, and this was actually after the masterclass, after intensive masterclass, and people still look happy. And so I hope we can see that this year as well. So during the few last years, we've done 30 masterclasses. We started in the Netherlands, but we've seen also that there's quite some international interest. So that's why we went to 6 countries to do these masterclasses and helped over 245 scientists. And I think this year we will also have quite an international amount of participants. So it's also good for networking and getting to know each other. So this masterclass, what do we do? Actually, before the start of the masterclass, you will receive access to VitroGo, our digital education platform. And in this platform, you can learn all about the what's, whys, and the hows of organ on a chip. So what is an organ on a chip? What is a chip? What is a pump? So learn about that theoretical part and why you would want to use organ on a chip, for which research questions, what can you do with it? And ultimately, how do you use it? So how do you choose a system? What type of chip do you need? What type of pumping system? So we go through all of these, and we try to keep it a bit interactive by using examples from the field, and also tickling your brain with quizzes in between. And we also provide a lot of resources and tools to use also in the lab to design your organ on a chip, and also look for what's out there, what kind of technologies are available. And then at the day 1 of the masterclass, we start with a learn and design challenge, so to say. This helps you to go from an idea or a research question all the way to setting up an organ on a chip assay. And we guide you through this process, which is kind of a 3-step design process. We start with defining an application and requirements. And then we let you get inspired throughout the masterclass to think about available technologies, what's possible, and yeah, to see what's out there, both in the industry, but also in academic examples. And eventually you will design your own essay, and also we will end with a pitch. So you will do this within groups, and usually this is a very well valued part of the masterclass. So we start with that, and then we will continue with another very valuable part, which is intestinal organoid virtual training held by Julie Watson from STEMCELL Technologies. And then we will continue with hands-on part, where we look at chips and flow. So we have different types of chips and different types of flow systems. So you get to recognize and learn to work with all these different types of chips and pumps, and also learn what the advantages and disadvantages are of some of them. And we will also look at mimicking seeding cells. And speaking of cells, on day 2, we will actually work with living cells, where we can learn on how to seed cells on an organ on a chip, on different types of chips, how to make sure that they stay attached and that they can grow well to be able to be perfused using an organ on a chip system. And then we continue with commercial demos. So we have also there a bit of hands-on experience for this commercial part. You will see all the available technologies from different companies, which will also help you in designing your own essay. And these are companies for this masterclass: Microlfuidic ChipShop, Proefdiervrij and Dynamic 42 will present their technologies or show about their activity. So you can also get to know them. And day 2, we will end with a networking session. So people from around Utrecht, but also with your peers, you can basically network with chips and drinks. And then the last day, we will have expert views. So we invited 2 experts, Professor Broersen from the University of Twente and NISO Food Research. She will talk about the gut and gut microbiome and gut-brain axis models. And for this, she uses organ on a chip models, and she will give examples from the field on that. And then we have Dr. Jean-Philippe Frimat, and he's a senior scientist from LUMC, Leids Universitair Medisch Centrum. And he has over 15 years of experience with organ on a chip, and he's a bit more from the engineering perspective. And he has most experience on brain and neurons on a chip, and he can tell you all about the tips and tricks and all the hassles you might come across in the lab with organ on a chip. So these experts will give like a real life experience and share with you. And then we end the masterclass with pitching your own designs. And here we can then talk about all the available experts that can give feedback and have lively discussions on the different types of essays that you've come up with. And if it's up to us, it doesn't end there, because we believe that this is a way to kickstart or to overcome limitations in organ on a chip. But of course, it's a longer process, so we will be there for continuous support if you need that. If you're not able to join the masterclass, I mentioned in the beginning, we have VitroGo, our educational platform. So here we have all the theoretical part, so you can learn, get inspired, and we have protocols, tools for designing your own essay, and also monthly personal assistance. So you can also alternatively just subscribe for that. You can also check our website for more information on this. And that brings me to the end of this part. So I hope you got a nice overview of the masterclass, and hope to see you there. And then enjoy the rest of the meetup.
Thank you very much, Emre. It was great. So I would like to ask Naya Moutsatsou from ICAT to tell us a bit more about ICAT. And if there are, of course, any questions, please leave your questions in Q&A or chat. We would be happy to answer them. Naya, floor is yours.
Thank you. Let me share my screen. Hello from me. I'm from ICAT. My name is Naya. You should be able to see my screen now, right?
It's coming. Yes, we see it now.
Yes. Nice. So ICAT is a part of the University Medical Center of Utrecht, and we're also part of RegMedXP, which is accelerating regenerative therapies. We're one of the pilot plants of RegMedXP. Our mission is to build bridges between research and clinical practice by uniting expertise and infrastructure to facilitate the translation of patient-specific advanced therapies. ICAT has 3 main facilities, the Open Innovation Lab, the GMP Simulation Facility, and the GMP Production Facility. And across these 3 facilities, we can enable all the process from early stage development of advanced therapeutics all the way down to GMP manufacturing of these therapies. So in our Open Innovation Lab, we do early research and development projects. We host academia, but also industry on their early research projects and across the fields of diagnostics, conventional therapeutics, biologics, advanced in vitro models, and advanced therapies. And specifically for the advanced therapies field, we have more expertise, so we can take these projects down the pipeline to GMP simulation and GMP production. So anything that has to do with cell, gene, or tissue therapies, we can move forward with GMP simulation and produce them as well. So the Open Innovation Lab is located now at Zeist, at the Utrecht Science Park. Next year we will move to the main campus of Utrecht Science Park, and is basically a turnkey facility with modern equipment for R&D, and we can host projects from academia, from startups, and from scale-ups as well. Is equipped laboratory and office space. We have a lot of advanced equipment on biofabrication, process development, and all this equipment is also compatible with our GMP production facility, so for further translation later on. We offer flexible memberships to the Open Innovation Lab users, and we are also very close to clinical expertise and leading experts of academia. Then in our GMP simulation facility, that's where all our expertise unites for ATMPs. So we can really bridge the gap between the preclinical research and the GMP production, which is often where we see a lot of gap. And we do this by employing quality control specialists, quality assurance specialists, regulatory affairs, but also technical expertise on gene editing, on viral vectors, on cell and gene production, therapy production, and on tissue engineering. So we can really bring in all this expertise early on into the projects. Then when a project is ready and ready to produce, has gone through the clinical or is going through clinical trials, and produce the material at our facility at UMC Utrecht. This is a 3 GMP Class B clean rooms at the moment, and we have another 6 being built at the moment. They should be ready at Q1 of 2026. This is a JC certified processing facility, an organ bank and tissue establishment, and of course it has GMP manufacturing license for ATMPs and also release of ATMP products. So at ICAT, we have a variety of projects across immuno-oncology, production of viral vectors, projects that deal with the musculoskeletal defects, with organoids, with diabetes, and they are spanning across early stage development, preclinical studies, but also we have projects that are in GMP manufacturing. So really across the whole pipeline. And our services, as I said, we do flexible memberships for our Open Innovation Lab. So startups or academic groups that spin out can rent equipped laboratory space, and they can do their proof of concept. Also, we do equipment rentals. So we have some specialized equipment that maybe a researcher does not have access to in their own lab, so they can use ours. We have consultation services on ATMPs, more specifically that deal with regulatory compliance, GMP compliance, health economics, quality assurance, and product development. We can also do contract research, again, more specifically to ATMPs. We can do feasibility and stability studies, process development and validation, and QC analytics development and validation. As I said, we have our own manufacturing clean room, so we can do the production of these advanced therapeutics there. And we organize also education and networking events like this one. So that's the core of what we do, let's say. We lately got funding for our own startup incubation program. So we partnered with Utrecht Think, is an incubator in Utrecht Science Park, and with funding from the province of Utrecht, we can now offer at very competitive prices our infrastructure to startups so that they can move their product development and they can take it to commercialization, and we can pair that with helping them with our in-house expertise and also with business development advice to really accelerate their go-to-market, basically. We have a vast network of collaborators, academia, non-profit organizations, but also industry. Yes, that's us. Happy to take any questions if you have.
Just right on time.
Good.
Thank you, Naya. Is there any questions? I don't see any questions. I don't see any questions in the chat or Q&A, and also no raising hands. So I will send your information, Naya, or any other emails if they have questions afterwards to everybody.
Sure.
But for now, maybe we can move to the next presenter. Here, and I'm presenting again. Great. Invite that while being in a conference, kind enough to join us and present technologies from Dynamic 42. The floor is yours, Tim.
All right, thank you very much, Hossein, for the nice intro. Indeed, been at the conference, but I'm very happy to introduce you into the Dynamic 42 organ-on-chip technology and focusing more specifically on intestine-on-chip applications. So my name is Tim. I'm an application specialist at Dynamic 42. We're quite a small industry startup, having now 25 members. We're based very centrally in Germany, in the small city of Jena, where also SAIS is located. We do all kinds of organ-on-chip services. So we sell our biochip products, but also the whole periphery around it. But also to pharma customers, they want to outreach their research or outsource their research. We can also do fee-for-service studies for them. And we're also involved in different kinds of hands-on courses, as the MasterClass is going to be one. All right, this is basically the heart of our whole organ-on-chip vision. So this is the biochip. Usually it's made from a biocompatible and low-absorbing chip material. So the chip material is PBT. It's non-PDMS, so it's very good, especially if you want to test small molecule drug testing for toxicity, particularly in the liver model. We designed the chip to be relatively user-friendly, so it's not a rocket science technology, so you can easily do pipetting in the chip without the risk of air bubbles. So we try to be as user-friendly as possible, and you can also attach the whole accessories around in a plug-and-play manner. And the nice feature is definitely that we have a relatively great price-to-performance ratio. And as you might already see, that the biochip is in a format that is compatible with imaging, particularly with live cell imaging, since it is provided in a microscopic slide format. All right, if you would now do a cross-section of the biochip, you can see that the biochip contains of two independent cultivation chambers. So we have a top channel here and the bottom channel here. And in between these two cell cultivation chambers, there is a porous membrane. And you can select, depending on your application, which kind of pore size you want us to integrate into the chip model. So we have a pore size from 0.4, 3, 5 to 8 micron. Particularly the 8 micron is really important if you want to investigate cell migration between the two cell channels, because the pores are large enough to let cells penetrate through the membrane. But if you want to restrict the cells to their designated channel, then 0.4 might be the better choice for you. And here on the biochip, you can see that it has two windows, and each window is the membrane area where the cells are cultivated. And these cell cultivation areas are not connected, so you have on one chip either a biological replicate or two independent samples where you can test your applications in. This is basically how the chip is connected to the pump system. So we have a pump head, that one here, that stays inside of the incubator. So your biochip and all kind of materials where the cells are in a humidified 37-degree environment. And the controller of the pump stays outside of the incubator so that most of the electronic part, which is very sensitive to the humidity inside of the incubator, stays outside. We have a circulatory perfusion loop, so we attach usually these microfluidic reservoirs here and fill them up with cell-specific medium. And then we perfuse in the direction here from the microfluidic reservoirs into our chip channel, where the cells sit on the membrane, and then the medium gets recirculated. Usually we use silicone tubing, which works very well for us, and this tubing can be easily attached into these cassettes and can then be clicked in a plug-and-play manner in the pump head. And the whole story behind the perfusion is basically that it simulates kind of a blood flow by constantly perfusing medium of the cells and providing also continuous nutrient and oxygen supply. And we are not only limited to perfusing one channel, you can also perfuse both channels of the chip at the same time. This is basically how it looks. So if the medium would now perfuse through the chip channel, you would see that you have your two cell types on each side of the membrane, and the fluid is going through the chip channel and is recirculated afterwards. All right, we provide all these kind of chips, materials, and pump systems also in a kit, as I mentioned already. So if you're interested in that, feel free to reach out. We will also launch a tumor microenvironment soon, but I know the session is more focused on intestine on chip, so I will go a bit deeper into that topic. So you might ask now, okay, chip sounds very nice, but what can I actually do with it? So basically, you're not restricted to any kind of assay or readout option that you know from conventional two-dimensional in vitro models. So the sampling of the supernatant from both sides is possible at all times during your experiment. So you can measure clinical parameters, release of cytotoxic parameters, for example, damage markers such as LDH. You can measure cytokines or compound turnover and a lot of different things that you're interested from the supernatant. But you can also recover the tissue from the chip by excising the membrane with a sharp scalpel, and you can either lyse the tissue to isolate RNA or DNA for downstream analysis, such as RNA-seq, for example. But you can also stain the tissue, you fix it before, and then you can go under a microscope and check for immunofluorescent markers. But also FACS is definitely an option that is possible. As I mentioned already, laser imaging is a possibility since it's a microscopic slide format. You just stain inside of your chip, you just put the chip under a fluorescence microscope, and you can check your live imaging dyes. And already, as you know, for enzyme activity, there are various assay kits available on the market, which are also compatible to the chip system that we provide. All right, we have all kinds of different established organ models at Dynamic 42. So we have established models of the liver, of the intestine, lung, and cancer, but also the vasculature only. I will focus mainly on two applications of our intestine on chip. But if you're interested in some custom organ model development, we're also happy to provide you with some more technical info about that. All right, this is just a showcase how our T42 intestine on chip works. So as I mentioned already, we have a two-channel system, and in between there's a porous membrane. Usually we seed in our endothelial cells in the top compartment, and then we integrate also resident macrophages here in the top channel. On the other hand, we integrate intestinal epithelial cells on the other channel, and then we perfuse both channels. And what the perfusion actually does to the intestinal epithelial cells is that under static conditions, the intestinal epithelial cells remain mainly flat. So they are flat, uniform monolayer in the transwall. But if you put the chips on the perfusion, you can see that they grow out to really nice three-dimensional architecture or morphology with this villous and curved-like structures. And this is also reflected here. So under static culture, the cells are very uniform and have a less microvilli density, but under perfused conditions, you really have the three-dimensional architecture with a lot of high-density villus-like structures. And we can also measure the barrier integrity by putting in a physiodextrin permeable dye. So we put in here in the intestinal side the dye and let it perfuse through the other side and measure the fluorescence absorption of the dye here. The more go through the barrier, the less intact is the barrier. But we can see that the perfusion increases the barrier integrity compared to static conditions. These are just some nice immunofluorescent images to showcase you that the tissue is viable, that we have a really nice confluent layer of endothelial cells expressing endothelial cell-specific markers, and here are our integrated macrophages. And we can also see the three-dimensional structure here, and the intestinal epithelial cells are really showing nice and compact apical cell junction complexes. But the intestinal tissue is not only very intact, the barrier, but the cells also transdifferentiate into specific cell types of the intestine, such as mucus-secreting goblet cells or also Panus cells. And also the cells show a relatively moderate amount of metabolic enzyme activity. So it's called CYP enzyme activity. So you might now ask, what applications can you actually do with the intestinal chip? So there are a lot of different applications that you can do with it, starting by toxicological assessment. So if you have a drug that you know is toxic to your cells, you can also test that in the intestinal chip, and you're not limited only to intestinal damage, but also vascular damage. So this is very nice. Disease modeling is nowadays very important for the intestine, since a lot of diseases such as inflammatory bowel disease or celiac disease are having rising incidences nowadays. So this is a really hot topic in the field of intestinal research. We can also do nutritional studies or drug delivery of different compounds such as nanoparticles and measure their oral absorption through the epithelial layer and also their distribution in the whole tissue itself. And another point I will definitely focus on is microbiota host interactions and also infectious diseases. So this is also another very hot topic similar to disease modeling. So the first one I'm going to start is the investigation of fungal host interactions. So we published this last year. We basically inoculated fungus, which is commonly not harmful for the intestine, into our intestinal channel here. So the fungus we inoculated was Candida albicans. And we put the system under perfusion. And what we can see is that in our chip model, the fungus really forms invasive microcolonies. And this is shown here. So you already see on the membrane that Candida albicans is forming these little dots. And we performed different kinds of readouts I already outlined. So we did IS staining, measurement of barrier integrity, but also quantified colony forming units from different compartments, so from the supernatants, but also from the tissue lysate, and also collected the supernatants for cytokine analysis. And what we can actually see here very nicely in this video is not only the three-dimensional structure of the intestinal cells, which are stained here by green, F-actin, but we can see in red that Candida albicans is forming these spreading microcolonies all across the epithelial tissue. And if I maybe can restart the video, we see that most of the microcolonies stay on top of the epithelial cells. And then in the middle there, you already now see the membrane with the pores. And you can also see that some of the microcolony particles are already infiltrating the vasculature, mimicking a dissemination, systemic dissemination of the fungus. So the fungus is really transmigrating through the membrane, through the epithelial tissue, into the systemic endothelial compartment. And we went then a step further and tested also antifungal treatment. So usually fungal infections are treated with antifungal administration, and usually this is done by intravenous injection into humans. So we put our antifungals in the vascular perfusion, mimicking the intravenous administration route. And we did this for both antifungal sensitive wild type strain and the resistant clinical isolate of Candida albicans. And we could really demonstrate differences between these two strains. At 4 microgram per milliliter, caspofungin completely killed the wild type strain in the model, whereas the resistant strain was really growing a lot into the microcolonies. So this is also dose concentrations that are usually administered in human patients. And we also compared intestinal fungal burden, so the CFUs that were visible in the lysate of the intestinal epithelial cells, so the candida that already migrated into the cells or invaded the cells. This was also shown with the fungal invasion here. And the dissemination, we measured the CFUs in the vascular supernatant, meaning the fungus that was translocating into the vascularity. And we can see in the red here with the red bars, this is the wild type, and it's decreasing with increasing concentrations of caspofungin. So it's really sensitive to the caspofungin, and we were really able to control the infection. However, the resistant strain is staying up on a level even at very high concentrations of caspofungin. It was only effective for the dissemination of the fungus. However, the invasion was still really high for the clinical isolate. And we were also able to measure increased release of IL-8 upon the infection with both the wild type strain and the clinical isolate, whereas caspofungin reduced it. Another application I want to show is the modeling of inflammatory bowel disease. So it's an inflammatory disease of the intestine, and we usually put in a trigger to disrupt the epithelial barrier integrity. And we put on also LPS in the epithelial channel to mimic the presence of bacteria. And when the epithelial barrier gets leaky, the macrophages in the system, they're able to sense the LPS gradient and then react with a chronic inflammation or excessive inflammation. And we were able to show that this IBD phenotype is really disrupting the integrity of the epithelial cells, but also the vascular cells. So the apical junctional complexes such as E-caterin and VE-caterin are really reduced during this IBD-like phenotype, and that the morphology was also depressed. We also measured a higher intestinal permeability when we induced these IBD conditions, and we were able to see a reduced intestinal height of the villus-like structures. So the 3D morphology was also affected, as it was shown here in the bright field images. And the treatment does not affect viability, so it was really an inflammation-mediated effect that we could show here. Definitely, we also measured cytokines from both levels. I do not want to bore you with too many scientific data, but what we could show is that in both the vascularity, we were able to show an increased release of the pro-inflammatory cytokines, mainly TNF-alpha and IL-8, but also in the intestine, we could measure elevated levels of pro-inflammatory cytokines in these IBD-on-chip models. A cool feature is also when you stain the tissue and you put them under fluorescence microscope, you're also able to do three-dimensional reconstructions. So you can really discriminate, okay, where is my tissue actually located? And we were really interested in the macrophages, so the immune cells in the system. And what we've shown here in the control, that the macrophages, they line up really on top of the endothelial cells above the membrane. And if we have these IBD conditions, the macrophages, they really start to actively migrate into the epithelial tissue. And this is also shown here in an orthogonal projection. You see really nice three-dimensional structure of the intestinal epithelial cells, and that under IBD conditions, the macrophages are really able to integrate here in the spots where the apical junctional complexes are not functional anymore. So you see it here in green that the macrophages are really actively infiltrating into the tissue. All right, just to conclude the applications, this is another very short overview about what we can actually do with multi-organ modeling. So we published this year a study by connecting gut, liver, and placenta. So it's already a 3-organ model, very complex organ model, where you can study actually the distribution and toxicity of some drugs. So this was published this year. But this is not restricted to only these organs. You can also connect only gut and liver or lung and liver. So this is something we are also further developing and advertising to people to use. And just to conclude, so in the future development, I think this will come out by the end of November or starting of December. We're currently developing the integration of tear sensors for the measurement of barrier integrity within our system, but also the integration of oxygen measurement spots. So these little sensor spots, which can be read out by a fireplate using a specialized software. And with these two measurements, we complement our portfolio by enabling live measurement of different parameters. So on the left side, tear for barrier measurement, but on the right side, also oxygen measurement. And with this, I would like to conclude. Thank you very much for your attention, and feel free to ask any kind of questions. Thank you very much.
Amazing, Tim. Thank you very much for the presentation. Maybe you can keep your presentation
Yeah,
there, just
sure.
in case if there's any questions there. Just a remark that we are going to have a general organ on a chip masterclass. Of course, team presented intestine on a chip, but they have also other models. And during the
Yes,
masterclass, you can ask questions about other models as well.
definitely.
So I will start with a question in the chat. There's the first question. Do you have chip with organ tissues from all the species, including human? This is actually a very common question.
Yeah, so basically all our models are based on human cells. So we're trying to avoid the use of animal models and also the use of animal-derived cells. I think it makes a lot of sense for toxicological assessment to compare also animal-derived cells in comparison to human cells, really benchmark, okay, this is really a human-specific effect, or this is only seen in animal models or in animal-derived cells. But we try to stick to human cells since our mission is to reduce animal models, so we do not want animal models to have in our lab, to be honest. But if a customer really is inquiring this, and we definitely do also the benchmarking between these two types. Yeah.
Yeah, and I can imagine if there is a cell line from animals, you can always replace it with, for example, cockatoo, and
For
it's a matter
sure.
of changing cells.
Yeah.
And then the next question from the same person is, have you evaluated immunology, efficacy, and safety of vaccines?
Okay, for vaccines, we do not have applications at the moment, but I can definitely imagine doing this. But then you might need a longer term model, a model that is stable for a longer period of time, since you have to target your immune cells to really function as memory cells and also to respond to the vaccine. So this is something we're not having at the moment, but feel free to reach out, and I think we find a solution where we can maybe also model this in our chip platform.
Yeah, perfect. Thank you. More questions coming. We have to use a scaffold to grow the cells in the chip or organize. So if somebody wants to use the scaffolds, I can imagine there are like fibrous scaffolds, they're self-standing or hydrogels. Can you explain a bit more about using them in the chip?
Yes, so basically for organoids and spheroids particularly, we also have a new chip version, which is called BC003. I think you can Google that on our website. It has integrated microcavities in the membrane. So the membrane has like little, I would not say holes, but really own cavities up to 25 per membrane, where you can retain the three-dimensional structure of the spheroid and organoid. So you load them as they are in their three-dimensional configurator into the chip, and you can also overlay them on top with the gel, so you can really mimic the extracellular environment of these cellular components. And the cool thing about this chip is that it not has only two channels and one membrane, it has actually three channels and two membranes. So you have the microcavity membrane, and on top you also have a membrane where you can also include a vasculature. And the thing with like putting a gel on top of your spheroids is that this channel might not be available for perfusion, since the gel is blocking the channel for a good perfusion. So we tried to include another channel to feed the spheroids from below. So this is definitely a thing that is possible, and I strongly advertise this chip model to use for spheroids and organoids.
Yeah, and I think this aligns with the next question, that can we add ECM on a chip?
Yeah, for sure. So usually we coat our membranes with normal collagen. So collagen 1 works for a lot of applications, but for some specific cell types, you also need collagen 4, some mixtures of laminin and collagen. Matrigel is definitely an option with different kinds of concentration. I think if you're already familiar with what kind of ECM your cell types need, then it should be easy to adopt it to the chip.
Yeah. And the good thing about this three-channel chip is that in one of them you can inject ECM. The channels are injectable, and then you can access the ECM from two sides, and there's a membrane in it. So there is another question. Can a steroid on a chip also be used for tomography?
Sorry again, didn't get that one.
Tomography. I'm not sure actually what can a spheroid on chip also be used for tomography?
Tomography, I'm not quite sure whether I'm correct, but maybe it means the scanning through the whole spheroid, so getting the whole three-dimensional imaging of the spheroid. So we developed also a clearing protocol for this, so to clear the spheroid from all kind of gel residues, but also from extracellular components that the tumor-associated fibroblasts around the shell are generating. So we're really able to also go into the core of the spheroid. So we usually do Z stacks in our fluorescence microscope, and I think that should also work for confocal or other kinds of fluorescent microscopes, where we can really do a 3D reconstruction of the spheroid. So this is possible, yeah. If you mean that by tomography, I'm sorry if
I think that's the question, and I want to add that you have a very thin bottom layer as well, right?
Yeah.
And then
and we can also extract the spheroids from the chip, so we can put them back in a well plate, fix them, clear them, and then should be easy to image them.
Yeah. I'm an independent trainer, and I think that's a very good feature of your chip, that you can take out the tissue. Yeah. And the next question is a very relevant one. For how long can you keep the cells in culture? Up to weeks or also months? And my question, adding to that, what's the longest you have kept the cells alive?
Yeah, very good question indeed. It depends very much on the cellular model you're building up. Usually we're having a culture period of 1 up to 2 weeks, depending on the model that we're using. And the longest one is actually our lung on chip model. So we can keep that for up to 5 to 6 weeks, because the lung chip will be exposed to air-liquid interface. So we airlift, so we remove every kind of fluid on top of the epithelial cells, so they stay in contact with air, which is normal for human lung cells, that they're exposed to air all the time. And we perfuse the models with air afterwards. So not with medium, but with air. And to differentiate the cells, that they also expose ciliae, so these moving particles on top of the cells, which remove the mucus or also clear the lungs from extracellular particles. This takes 5 to 6 weeks.
Yeah. More questions coming. I'm not gonna stop.
Yeah, no worries.
Unless you tell me, Tima.
Yeah, it's good that people ask so many questions. I like that.
Yeah, because it's interesting. So the next question, I think we covered a bit. Can we use hydrogel-based ECM to culture cells, and how we will be able to characterize them?
Yeah, this is basically what I mentioned. Yeah. And if you want to characterize them, try to solubilize the gel or remove the gel by clearing, or you can also extract the cells from the other side of the membrane if your cells are not really suspended in the gel.
Yeah. Now also, I think it depends what they mean with chemical characterization, mechanical characterization.
Yeah.
Yeah. Okay, and the next one is, what are the possible ways to sterilize those ECM material?
The chip material?
ECM material. Maybe you can explain. ECM is a bit...
Yeah. Okay, I try to answer both. So chips can be sterilized by 70% ethanol. You just immerse them or submerge them into the ethanol and pipe it through. And afterwards you wash with water, and then you're good to go. And for ECM, usually it should come sterile from the vendor. So we use only sterile substances and medium and cellular components and put them in the chip. So usually our ECM that we use is completely sterile. And to get sure, we also sometimes put antibiotics in the medium for the cells, so that contamination is nearly impossible.
Yeah. And if you want to explain a bit more about ECM sterilization, because usually you get the ECM sterile, you can ask more questions in the chat. Next question, in conventional adherent cell culture, we need to passage on the basis of confluency. How does it work in the case of organ-on-chip platform, in your platform, for example?
Yeah, so basically organ-on-chip is seen as an endpoint, so you do not passage in the chip. So basically you would have a pre-culture of your cells, and usually you would split or detach them if they're subconfluent, so not 100%, maybe like, I don't know, 70 to to 85% or 75% confluency, and then you seed them in the chip. And usually in the chip, they need to be confluent because otherwise the perfusion might remove the cells because there's a lot of space where, or empty space where the perfusion can detach the cells. So they really need to be confluent in the chip before you're putting on the perfusion.
Yeah, yeah. And usually organs on chips, because of the size, they are not used for expansion.
Yeah, yeah. That's a good
Next
remark.
question. How can you control the barrier integrity, tight junction tier, and shear stress for intestinal model?
Yeah, for the intestine, I think I mentioned it a little bit. Maybe it was not super clear. So if you want to do an endpoint determination, then the FITZ-dextran assay is the most common, I would say. So FITZ-dextran is a permeable tracer dye that you put in one channel. Then, for example, you turn the chip upside down and let it diffuse through the barrier. And the more tight the barrier is, the less of the dye is found in the other channel. If the barrier is really leaky, a lot of this dye will be found in the other channel, and you're able to quantify this in a spectrometer. So you just measure the absorbance of the dye. So you can just put it in a microplate reader and measure at a specific wavelength. The other method I presented at the end, so the tier measurement is an electrical measurement, and this measures the barrier integrity also over the cell surface, but it is a live monitoring. So you can really measure every 10 minutes, every 20 minutes, every 30 minutes how intact the barrier is. So this is definitely a plus compared to the endpoint assay using FITZ-dextran. But this will be available by the end of November.
Yeah. And I think you just briefly showed in your presentation that because the question is also about controlling the barrier integrity, and we know at least in the intestinal cell model, if you add flow, barrier integrity goes higher. You can see also in the graph as well.
Yeah, that's true. And especially it's really cool in comparison also to the oxygen monitoring. So these two complement really for microbiota co-culture, because if you have microbiota inside, you really want to get sure, okay, your model is still stable, that the microbes are not overgrowing your whole tissue. And this you can determine by a measuring of barrier function and also the oxygen content. Yeah.
Yeah, very good point. And then the next question is, can you subculture cells from the chip?
This might be the same question as the expansion question, right? So I would maybe skip that. So this is, I think we explained that, right?
Yeah, at least it's not common.
Yeah.
Right. And then it's a follow-up of question about ECM sterilization. I'm not sure if you can answer this, but for example, if it's a decellularized ACM extracted from tissue sample, how can we sterilize them and add them to the chip? Do you have experience with that?
To be honest, not really. I have some experience with human placenta-derived collagen. So they extract the collagen from human placenta, and I know that they sterilize the collagen by plasma treatment. It's either plasma or ethane oxide or something like this, like gas sterilization. I think either one of these two, I'm not quite sure, but they usually sterilize the extracted ECM before they ship it. Yeah.
Yeah. Then the next question would be, is it possible to collect the media later on to test if the organoids secrete some components, for example, growth factors?
For sure. So you have the medium reservoirs where you can just extract the medium supernatant on a daily basis, or if you want, every 12 hours. Usually when we're not doing the assay on the same day, we just keep the samples, spin them down, remove all kind of cellular debris in the medium, and then we freeze them at -80 or even lower. And then afterwards just thaw the samples again and use them for your assays. So all kinds of assays possible, cytokine measurement, viability assays, so or secreted factors, as you mentioned, for the organoids.
Yeah, perfect. Okay. I'm afraid we are gonna have to stop here because we are like 12 minutes over time. Thank you very much, Tim. If you could answer the questions in the chat if you have time, and also add, because we talked about this 3-channel chip, you can add a link to that chip. It would be great so people can go and study more. And you're going to get more information, an email address from Dynamic 42. If you have more questions, you can ask them later on after the webinar as well. Okay, thank you very much.
Since I gotta leave, I think it would be good if you directly shoot me an email if you have more questions. So I will also put that in the chat. Yeah.
Perfect.
But thank you very much for your attention, guys. It was really good.
Thank you very much, Tim. Thanks a lot. Okay, time. I'm sorry, we have to move on. Thank you, Tim, again. And the next presentation is by Lisanne de Vor from Proefdiervrij. Lisanne, are you here? Yes.
Yes, I am. Let me switch screens so you can see my slides.
Yeah, you can see it.
Thanks. Yeah, it's great to give a talk here in behalf of Proefdiervrij. We are a Dutch NGO, and we are working on making the transition towards animal-free science possible. So today I will give you a short introduction about what we do and how we see the future. So just a bit about me. I'm Lisanne de Vor. This is me in the lab. I look a bit different, but you have to trust me that it's really me. I am actually trained in Utrecht, where the MasterClass is also being held. And I did a PhD in infection and immunity, where I also had to work with animals. And because of that, and because it never really felt good, I came in touch with Proefdiervrij. And after my PhD, I started working for Proefdiervrij. And our goal as Proefdiervrij is to make animal testing obsolete. So actually we just have good alternatives and we don't need it anymore. We are working towards a future where we can make that goal possible. And for most of us at Proefdiervrij, it's also because of ethical reasons that we think that we shouldn't put animal lives below human lives. We also really think that using human-centered models makes science better. And then better not only in the sense of better translatable to humans, but also better as in more ethical. To show you that we really think it can make science better, I will go over a few numbers with you. So in 2023, those are the most recent numbers for the Netherlands. There were a bit more than 400,000 animal tests registered in the Netherlands. There is more behind this. So also because to make an animal experiment possible, you have to breed animals, and those animals that are parent animals or that are not the right age anymore or the right sex, they are not used in an experiment, so they are not counted. But there are also a lot of invisible animals used in experiments, so in the lab without even seeing the animal. So then you can think of FCS or using matrigel for organoid culture, but also the use of polyclonal antibodies that were raised in animals. And we use those a lot in different assays. So there are more animals used in science than only that number that I just mentioned. And just another number. Of course, not all animal tests are being done to test drugs. But it really makes clear that animal testing does not always really translates to the clinic. So 90% of drugs that seem to be working in animals actually fail in clinical trials, and that's quite a frustrating number. And we are really convinced that we can do better as scientists. And of course, there can be multiple reasons that clinical trials fail. Of course, it also depends on the inclusion of patients, for example, or strategic reasons of the company. but 70% of this 90% is actually because of efficacy reasons or unpredicted side effects. And that really shows us that our preclinical research is it can be a lot better, and animal work is a big part of that. So yeah, we always say lab animals are not humans; they are just a model, and because it also brings along a lot of animal suffering, we really want scientists to move to human-centered models. And luckily a lot of scientists also, you are also interested in using these models. For example, organs on a chip or organoids or in silico models, but we also think about a really simple solution, just use actual human tissue that is left over from surgery, for example. And we see that a growing group of scientists is willing to work with these methods, and they also think that they are more relevant than animal methods. But yeah, despite this growing interest in human-based methods, we also still see that animal methods are still the golden standard, and we really want to change that. So just to say that again, we don't want animal work to stop immediately without there being good alternatives, but we want the alternatives to become better and better, so that scientists are choosing these alternatives because they are the best option. But yeah, so the golden standard is still animal work, and that we see in the regulatory part. So it takes a long time to take up animal-free alternatives in the guidelines for safety testing, for example. And it also takes a long time to remove the animal experiment from these guidelines. And in the lecture that we will give in the masterclass, I will give you an example of this. And for animal testing, just for fundamental research, for example, we also see that we are dealing with a great animal methods bias. This, in short, means that reviewers are asking for animal experiments, but it's also often much easier to get funding if you are including an animal method in your application. And we will discuss this also in the lecture. It also means, of course, that this is a systemic problem, and there are multiple stakeholders involved. So it's not only you as a scientist who can make the difference. This is why we always say that cooperation is key. And we are working together with different stakeholders. So we are working together with the government, scientists, but we also like to inform the general public, and we are working together with other NGOs, just to make sure that we are making a change on all these levels. So what are we actually doing about it? You might know us from demonstrating on the streets up until 10 years ago, which I think it was useful back in those days, but now we changed our strategy 10 years ago to collaborate with scientists instead of working against them, because we really think that this makes reaching our goal a lot easier. So what we do is we are raising awareness in the general public. So we're quite active on social media, where we get sometimes a lot of comments if we post about why animal experiments are done. Then the general public sometimes responds that it's crazy that this is still happening, and that scientists should test on themselves or on prisoners, for example. And then, of course, we always respond that it's a system, and that scientists are not doing this for fun, and that it's important to make alternatives, but they are still part of a system that they cannot change. So we really want the general public to know this, and to not blame the scientists for this. We also publish articles in newspapers, and for example, for the weekend of science, this is more to a weekend where parents can go with their kids to the lab to show them how science is done. And then we made a board game to explain the use of animals in science, and then they had to save lab animals from the lab and replace them with alternatives. So that's what we do to inform the general public. We also award promising scientists that work on animal-free methods. We organize a yearly venture challenge. This is a sort of a boot camp where scientists can work on their business plan to make a startup. We often see that these alternatives are developed at universities, but then it stays there. It's not being developed more and more into a commercial product, or it's not being implemented in industry. And this way we want to change that. And in the Venture Challenge, different teams are competing with each other, but also collaborating in writing a good business plan. And they are discussing with bio-business entrepreneurs to make it better. I will also tell you more about that in the lecture. And then we are also a partner of the government. The government has this program called Transition Program to Animal-Free Innovations. And they are working on solutions to make this transition even faster. And we often go talk to them and think along in what we can do together. And then I think most important and good to know for you is that we also fund animal-free research, because we want to make it easier for scientists to make the step towards animal-free research. So we are sometimes part of a consortium for bigger calls. Then we add up to 100,000 to the project or to the grant. But on the other side, we also fund smaller grants up to 5,000 euros, and this doesn't seem so much. But you can think of an application for replacing FCS in your culture medium, or replacing polyclonal antibodies to make your experiments animal-free. And I will tell you more about the cons and the pros of making your science animal-free also in the lecture. But you can also think of using this grant to learn a new technique abroad that is not being applied in your lab yet. So we think it's really important to help scientists with also making these small steps. I'm keeping this a bit short, but we also, of course, are interested in collaborating with you. So you can choose, of course, what you can do is choose human-based research models. Well, you are already interested in this, otherwise you wouldn't be here. And I really wanted to show you that you can also apply for funding at our NGO. And during the lecture, I will give you more tips and hand you more initiatives that are being developed in the Netherlands and in Europe to help you work animal-free in your research. So I want to end with a world without animal testing is closer than you might think. And if you are already interested in collaborating or being updated on nice events that are linked to this this topic, then you can scan the QR code and join our newsletter where we keep you updated on nice scientific articles, progress that's being made in making the world lab animal free. And thank you for your attention. If there are already some questions, I'm happy to answer them.
Thank you, Lisanne. It was great to see you here, and I think we all have a common goal to help participants adopt technologies like organ-on-chips faster and more efficient. Choose the right model for the right question. There is one question. Are you funding other countries than EU?
It is possible that you can receive funding if you're not based in the Netherlands, but then your project has to be connected to a Dutch institution or a university. So we are focusing mainly on the Netherlands. But if you are interested, there are similar NGOs or organizations abroad, and we can help you find them.
Yeah. And also, if people are joining the masterclass, apart from the masterclass and the technical part, we can also look for funding opportunities, including the ones that Proefdiervrij supports with an organization in the Netherlands, and just match you with the right organization.
Yeah.
Yeah.
Good idea.
Okay. I don't see any other questions for sake of time.
Yeah.
Thank you, Lisanne.
Good.
And if there are more questions, they see the email address, your email address here, and they can also leave it in the chat as well.
Yes,
thanks a lot, Lisanne.
Yes, no problem.
So now it's time for another cool presentation from Microlfuidic ChipShop, Dr. Franziska Boelke. Are you here, Franziska?
Yeah, I'm here, Hossein. Hi.
Perfect. Floor is yours. Thanks for being here, Franziska.
Thank you. Let me share my screen with you. So, and let's start right away.
Yes,
so,
we see your screen
can you,
properly.
Okay.
Yeah.
Perfect. So, hi, everybody. My name is Franziska Boelke. I'm working as a scientist at Microlfuidic ChipShop in the Research and Development Department. So those of you who maybe never heard of us before, why I cannot go on here? Nice. Okay.
Maybe different screens or?
Oh no. Okay.
Oh, yeah.
Just to let, I'm so sorry. So, Microlfuidic ChipShop was founded in 2002 as a spin-off of the Fraunhofer Institute for Optics and Precision Engineering. We have approximately 100 employees currently, and we are located at the green heart of Thuringia in Jena, in one of our high-tech regions. We have about let's say 5,000 square meter area where we offer the whole fabrication structure to develop, design, manufacture polymer-based microfluidic systems and in vitro diagnostic, but we are not just focused on polymers, we also manufacture and design in silicons, metals, glass is upcoming popular. Yeah, so everybody needs a mission. So our mission is to shrink the biological and chemical laboratory and to bring the lab ownership systems in your daily laboratory life at a reasonable cost. So what are we do exactly? we let's say we are divided in different main areas. One huge area, the customized development of and manufacturing. So that means if customers come to us with an idea, we offer the design, the production, manufacturing, and the distinct materials, and up to integration of molecules, coatings, and so on. Second area are we also develop complete systems that include a diagnostic chip and an analysis instrument. Next part is also we run a lot of collaboration projects and funding projects with different research institutes. And last but not least, our off-shelf products. We working continuously on a growing portfolio of different chips and other accessory, which is let's say very, very useful and popular to customers and scientists who start with microfluidics to kind of perform a proof of principle at first. So to give you an overview today of our organ on a chip portfolio, I try to divide it in let's say parts. First of all, I call it simple chamber chips. So that means one chip on a slide format, which are available in different sizes, surfaces, materials to establish cell monolayers. Second part or area would be interconnected chambers where you can perform interaction studies. I will show you some examples later on. One bigger part and very popular part, we often use our membrane chips to perform barrier simulation and 3D models, co-cultivation, and many more. Let's focus on a few examples. I also want to show when we meet at the hands-on training in a few weeks. So let's start with the interaction chip. As you can see on the right-hand side, we offer different designs of interconnected chambers on one chip slide, where you can perform chemotaxis, migration assays, cell signaling and cell-cell communications, but also so the chip enables for multi-organ models. As you can see in the lower picture, you would have 3 compartments with one bigger chamber and 2 smaller chambers. You would be able to cycle the whole compartment, like you would introduce a cascade of liver cells and kidney cells, for example, and you can cycle it on its own or all together. That would be a possibility for interconnection chips. Another example would be this interconnection chamber chip with 5 units. One unit is divided in, let's say, 3 channels. One main channel is disrupted or connected to the side channels by pillars, and those pillars I lost what I wanted to say. Yeah, these pillars have different pitches, so we try to give the customer the possibility to play around what really suits to its experiment. Yeah, so far to the interaction chips. The next part would be our family of cross-flow membrane chips. So we started many years ago, let's say 10-15 years ago, created the first design of our cross-flow membrane chips. We offer these chips, which are formed by 2 chambers laying to each other, divided by a semi-permeable membrane. These colored parts describe these 3 areas here. In blue, the membrane, the lower chamber, and the upper chamber. One opportunity would be to integrate different kinds of membranes here. So we have different pore size, different pore densities, which also influence the light transmittance, like you can see it in the lower picture. As thicker and higher the pore density is, then the membrane looks more, let's say, whitish. We have a lot of different designs. And if you want to have a look in our catalog, which is available at our website, you can follow the pages 100, and we'll find a lot of design technical possibilities to perform cell co-cultures. Here you can see some examples, as I already described, very, let's say old design, but very popular, very suitable, is this fluidic number 480, where we also had some cooperations in the past, and where you can also find publications, for example, by Knut Rennert et al. Or Maurer et al. in biomaterials, which work with the ships and establish liver kidney cell co-culture models and intestine models on this ship design. And if you, let's say, play around a little more and interconnect the chambers, building cascades, you would get the second example, where you have 4 units, 4 chambers, 1 bigger one and 3 smaller ones. This is a development made in a funding project a few years ago, where they simulate multi-organ system, liver kidney system, and we also introduced interfaces for sensors for parameter monitoring, like CO2, oxygen, glutamate. We fabricated it in different standard formats we offer. So in this case, it's a double slide format. And the last example to in case of membrane chips, is a quite new design for the establishing of skin model. So what we've done here is to slightly change the heights of the different the lower and the upper chamber, because we need more space on top. And here you would have different possibilities. You may see the this part on top or at the one side of the chip. This is a separate foil, so you would get it in a like a set, and it's created as an open tape chip. So that means you would be able to directly bioprint your model on the membrane, and then close the chip by yourself, or close the chip at first and try to establish the whole model on chip. In case of bioprinting, we have also some other cooperations and projects running. Some time ago, we started with the integration of a hybrid PP printed scaffold for bone marrow simulation. And integrated a 1 millimeter thick scaffold into a chamber and closed the chip with a adhesive foil. And in that first case, that's it. So the chamber is closed, the chip's closed system, and it's, let's say, it's not impossible to reseal the chip again, but it is no fun. So what we do next was to manufacture a second model, what you can see in the right lower picture, the explosion view. So we create a thicker scaffold of 3 millimeter size, which simulate cartilage and bone. And here the goal was also to mechanically interact and actuate the cartilage and to trigger the integrated cells. And that is why we focused on a resealable system. That means we close the chip at the upper part with a soft polymer plug, and because the system is not the best for microscopy, you would be able to reseal and open the chip, take out the grown scaffold after the end of the experiment, fixate histology at further analysis. But our newest work was also we succeed in print directly into a closed chip system, which make things lot easier, and we are quite excited to go further with this. Maybe I will be able to give you some examples when we meet in 2 weeks. Let's have a look at a few other examples. So in my experience, it's not so long ago, or let's say
Sorry, Franziska, to interrupt.
it's two last. Yeah.
Yeah. Okay.
Yeah.
Thank
Yeah.
you then.
This is
Yeah.
the one of the last examples. So you may be also interested in, as I experienced it and at Corona, it was a lack of biomaterial from the hospitals, and so that is why some of our customers and project partners started to work with the creation of spheroids. So this is another example how you could cultivate or generate preformed or on a chip directly generated spheroids on chip. That is why we also try to focus more on coatings on which which focus on the anti-attachment of cells. And last would be a little excuse, let's say, not just focusing on adherent cells and cell lines or co-culturing, but also, and it is quite difficult because normally you would flush out the cells of on your channels, on your chambers, and that is why we also started to develop different designs, different chip possibilities to culture suspension cell lines. Thank you so much for your attention.
Thanks a lot, Franziska.
That's it for now.
Yeah, thanks. So if there are urgent questions, you can write them in the chat or Q&A. I just wanted to add here that Microlfuidic ChipShop has a big library of chips, but also different components that you can use in organ chip systems. And because in the masterclass we focus on your application first and choose the right system for that, it's really nice to have a big library like what Microlfuidic ChipShop has, and then choose from those technologies if necessary. So that's really cool. And that's what we are going to do in the masterclass as well. And then you're going to get a very good introduction from Franziska, which gives you a lot of ideas about how your system can look like. I don't see any other questions, but I want to thank you, Franziska, for being here, and I look forward to meeting you in 2 weeks in Utrecht.
Yeah, me too.
Thanks.
Thank you so much, Hossein. See you soon.
Of course. See you soon.
Bye.
And bye. And now we go to Julie Wasser from STEMCELL Technologies. Sorry, Julie, for waiting.
Oh, no worries. Can you hear me?
We can hear you.
Okay,
Yeah.
I suddenly an alarming message that my disabled would happen. That wasn't true. Thank you for having me.
Of course. You can share your screen. Thanks for being here.
Sure. All right. Well, hello, everyone. So slight change of topic now. So I think you should be able to see that, hopefully. So
yes.
What we're going to, great. So what we're going to talk about now is stem cell organoid media and training. And I'm a scientific support scientist in epithelial biology at STEMCELL Technologies. So to start off with, I just wanted to sort of position our offering alongside what you'll be learning about in the Organ on Chip workshop and some of the topics you will have heard about already today. So if we consider the different culture systems that are available nowadays, here are a few of the options. So you've got the submerged culture system, which is kind of the traditional cell culture system, so obviously simple and well established, but does lack that complexity that we want to see really nowadays. You've got air liquid interface systems, popular in the lung and intestinal field, and these can be used for studying barrier integrity and transport. They do require an insert. Then we have organoids, which is what I'll be talking about in more detail, and these really are more physiological than, say, your traditional 2D culture system because they contain different tissue stem cells. And they also on complexity, even model multi-tissue complexity with a chip if you have one cell type and one cell type. They're cheaper than animal models, and historically there's been a lack of standardization, although that is perhaps now emerging. And these can be used in a complementary way to organoids as sort of different work streams, or you can also incorporate organoids into certain types of chips, as we were just sort of hearing, basically. And then, of course, you've got animal models, which do capture the full physiological complexity, but are expensive and time-consuming and not human. And nowadays, that's less required in America and other counterparts around the world. So as I say, in at STEMCELL, what our expertise is really in organoid culture and perhaps air liquid interface culture. And these are the media that we sell to support organoid culture. So we have a range of media for tissue-derived organoids, and these include intestinal, hepatic, pancreatic, lung, fallopian tube, and then also some contract assay services. And then we also HSE-derived organoids, including an intestinal organoid kit, hepatic, kidney, quite an extensive neural range, airway and vascular. So quite a range. So hopefully this could be relevant for quite a few of you, just because of the breadth here. So what's nice about using our media is that it's much more straightforward than making it up yourself. As you may know, stem cell media are typically quite complex and contain many different components. So back in the sort of bad old days when I was training and we had to make these things up by hand, we just spent a huge medium, which is nowadays that's not necessary with these types of products. So because we make our media in big batches, it means that they're very standardized batch to batch. So they function quite consistently. If you make up these type of media in the lab yourself, you're typically having to collect conditioned media, for example, from certain types of cells, and it just does vary quite a lot batch to batch. So our media are complete, optimized, and quality controlled, which ultimately saves you time and makes your experiment and work better. So we have a question in the chat. Do we have myocardial stem cells? So we sell kits for culturing. So we sell media for culturing primary myocardial cells, and also for deriving them from pluripotent stem cells, both ventricular and atrial cardiomyocytes. But we don't sell the cells themselves. So by and large, we focus mainly on offering culture media, although we are starting to move more into offering cells as well. And we also have another question. Are you planning to extend the list of organoids in the future, for example, mammary organoids? So we do have a medium called Moutsatsou, which is suitable for growing mammary sort of solid steroids from primary tissue or from breast veins. It's an area of interest for R&D to generate a more sort of modern mammary organoid culture. So hopefully we'll do that. But again, yeah, we don't actually sell the organoids themselves, just the media. And just my slides. So as well as the media, we also offer a number of ancillary reagents which can be quite handy, for example, or ancillary materials. For example, this organoid culture plate. So if you consider a standard organoid culture in, say, a 24-well plate, you'll typically have a dome which contains the organoids, and then some medium which sits on top of it. And what you tend to see is that the organoids sort of grow around the edge of the matrigel dome, but you'll have like a dead zone in the center where the nutrients can't penetrate. So this means that your cultures can be a bit uneven, and that the size of the organoid, as well as sometimes observing death in the center of the dome. You also have a meniscus forming, as you do with any fluid, because of the surface tension as it reaches the plastic. So that can cause problems with imaging. So we've released a new organoid culture plate, which functions in a way, where you have the matrigel sitting in this kind of bucket in the bottom with the organoids and the matrix inside it. And that means that you don't have the dead zone. The organoids grow much more homogeneously, and because we have a step at the point where the liquid interfaces with the plastic, you also have no meniscus, so that's much nicer for imaging. Okay. And all of our products are backed by a robust quality management system, which means that we really do have very rigorous processes and documentation to support the manufacture of all of our products. And what can be quite handy on a practical level in the lab is that we do have detailed protocols and technical support. So I'm part of the technical support team, but there's like 40 of us around the world. So if you have any problems with your experiments or any questions, or if you want to do something that's a little bit unusual that isn't covered by our kind of written protocols, you can get in touch with us, and we offer very comprehensive support to our customers to assist with this kind of thing. So you can email us, or we can have a meeting or whatever. And we also have these resources available online at our website. So if you just go to www.stemcell.com/organoids, it will bring up all of this. And what's particularly nicely is the tech tips and protocols. So these will have protocols for things like how to stain organoids, how to dissociate them and put them into flow cytometry applications, you name it, all different kinds of technical tips and protocols. Some posters that we presented at conferences, and our training courses, which are also quite so we offer a range of different modalities when it comes to our training courses. So in person, virtual, like I'll be delivering as part of the masterclass in a couple of weeks, and then on demand, which means kind of pre-recorded videos online. And we offer these in a range of different topics. So in terms of organoids, we have human intestinal, mouse intestinal, human hepatic, pulmonary, and then various other subjects as well. And these are all available, the on-demand courses are all available on our website for free. So, yeah, so I will be joining all the other speakers you heard from to help out with ASAR's organoid chip master. And so I focus on in this session is how to establish intestinal organoids from primary to how to passage them, and then how to dissociate them and make monolayers or air liquid interface cultures. And I think that aspect in particular would be quite relevant to generating chips. So this will be very much an interactive session, so you can ask whatever questions you want. And you can also bring in your own research topics and any technical issues you're having in the lab, because that can be really interesting for the other participants to hear about as well, and we can talk those through in real time. And that is it. So yeah, I look forward to meeting with more of you in a couple of weeks. And I see we have one more question from Tatu in the chat saying, back to the new plate for growing organoids. Do all the organoids get the nutrients equally? Yeah, that's the idea, because the it's flat and the medium is just sitting on top of it, yeah, the organoids get the nutrients equally, which means they're all kind of equally healthy and are the same size, which brings more homogeneity to your cultures. Are there any other questions before I hand back to Hossein?
While we are waiting for more questions, just to mention that this masterclass is to help you understand whether organoid steroids are models for you, and if organoid is the answer, which organoid model can fit your questions, maybe in combination with organoids inside organoids chips. Yeah, there are more questions actually.
Definitely. So do we have skin models? Yep. So we launched a expansion a couple of years ago. It's called Dermacult. And that offers kind of very good support for of keratinocytes, so you can grow them for ever so many passages. I want to say about 30 passages from both fetal and adult tissue, and then you offer more products in the skin area. We have media for co-cultures. We have some protocols on our website for co-cultures. So, for example, we have well-established protocols intestinal organoids with mixing our intestinal organoid medium with the T-cell medium in a 50-50 ratio and growing them as a mixture. And we also have protocols, for example, for growing pulmonary airway cells, epithelial cells with macrophages and various other combinations. So if you have a particular combination that's thing is to reach out to myself or the tech support team, and then we can let you know if we have any particular suggestions.
Okay. I don't see any other questions. And we are 15 minutes over time. So I think we can thank you, Judy, for being here and for your presentation.
You're welcome.
And look forward to over 2 weeks and intestinal organoid training as part of the Organ-on-Chip Masterclass. Thanks again, Judy, for being here. Okay, great. I think this is the end of the webinar. Just wanted to emphasize that we have 1 week until the deadline of the registration. We have a few spots still left. If you have any questions, just feel free to let us know. We will send an email to all of you, also those who have registered but couldn't make it, with the link to the recording, with the information we provided here, but also email addresses and means that you can contact the presenters. And the last thing I wanted to mention that during the masterclass, we help you choose the right technology, also follow it up with you to have those technologies in your lab. It's only this fast to follow up afterwards already. Okay, thank you all very much, and thanks for being here. I hope you had a nice time in this 1.5 hours, and hopefully see you maybe in the masterclass, but also somewhere in the future. Have a nice day, all of you.